if you want to know how someone can dissect a basking shark that has washed ashore from a surfboard, you're going to want to listen and watch this episode, because we have Chris Mull on the episode who talks about his experience in dissecting a basking shark that washed up, unfortunately dead, but he didn't want to have it go to waste. He got a bunch of different parts so that it could be used for science. We're going to talk about that on today's episode of the Beyond Jaws podcast. Let's start the show. Hey, everybody. Welcome back to another exciting episode of the Beyond Jaws podcast. I'm your co-host, Andrew Lewin, with my co-host, Dr. David Ebert. David, imagine you pull up on a basking shark. You can't get out there right away. So you know what you do? You just take your surfboard and you go and dissect it. It sounds like an easy job, but it's not. We're going to find out about how hard that is. Imagine, what would you do in that situation?
Well, for everybody in context, it washed up on a little island that he had to take his surfboard out to get to collect stuff. Then he had to make multiple trips back, and he had the rising tide coming in or going out.
Fast in Nova Scotia. It's fast.
Yeah. And so he had a few challenges, but that seems to be, if you work on sharks at all, you're used to doing dissections and necropsies, that's not unusual in the field.
Exactly.
It have to be kind of challenging. So that was quite a story Chris had to tell, and I hope everyone listens to it too. It was
absolutely.
It was just one of many fascinating stories he had to tell about his journey today, and which his journey, as we'll find out, was he's driving across Canada right now with his wife, and that's turned into a bit of a journey in itself.
It sure has.
And we'll share a little of that too. So
But, it was a lot of fun. It was a lot of fun to interview Chris. He was really forthcoming with a lot of information with his career, where he started, where he's ending up, where he would like to end up, and I think it's really interesting to show that having a shark science career is not an easy path, and there's a lot of things that you can put yourself in a good place, and hopefully people will see it and put. And give back in terms of show hey, we've got love for you and you deserve to be in a certain place. And I think that's what we see with Chris in this episode. So Dave, what do you say? Should we get to it?
Let's do it.
All right. Here's the interview with Chris Mull talking about his career in shark science. Enjoy the interview and we'll talk to you after. Hey, Chris, welcome to the Beyond Jaws podcast. Are you ready to talk about sharks?
I spend most of my time talking about sharks, so absolutely, I'm ready.
Wonderful. Love it.
Great. That's like the hear. Well, welcome to the Beyond Jaws podcast, everyone. And we have an outstanding early career guest today. We have Dr. Chris Mull, who's currently, among other positions, is a research scientist at the University of Washington School of Aquatic and Fisheries Sciences. He also holds several postdoctoral positions we'll talk more about today. Chris is originally from Southern California, where, like most Southern Californians, he spent his time at the beach, surfing, snorkeling, diving. Think Baywatch for shark nerds. And he did his undergraduate, though, at the University of California, Davis, up in the Northern California area, where he got his first experience doing animal husbandry and research, working on everything from crayfish to sturgeon to sharks. After his undergraduate, he moved back to Southern California to do a master's degree at California State University, Long Beach, with Chris Lowe, who's a frequent podcast guest, and Chris was recently on our Jaws 50th anniversary episode. Mm-hmm. And where he worked on the reproductive biology of those lovable flat sharks, the famous round stingray Eurybates halleri, who we'll find out more about today. After his master's thesis, he worked at the CSU Long Beach lab as an analytical chemist doing toxicology just for fun. He then moved to Vancouver, British Columbia, where he worked with Dr. Nick Dulvey, to do his PhD. And Nick has been on our podcast as well back in October of 2023. And also on his committee was another fan favorite of ours, Dr. Carrie Yopat, who's also been on our podcast. And we really need to have her back again sometime, studying the life history and brain evolution across chondrichthyans. After completing his PhD, Chris did several postdoctorates in the U.S., Australia, Canada, and his current work combines phylogenetic comparative methods to better understand trait evolution and more applied work looking at how international trade contributes to unsustainable fisheries for sharks and rays. Chris has over 20 publications, and he's also an associate editor for the Journal of Marine Biology. As I mentioned, he has a research position at the University of Washington, has several postdocs. He's also the co-founder and principal investigator of Sharkopedia, which we'll find out more about today. He's also a principal investigator for the National Center for Ecological Analysis and Synthesis to assess international trade that drives unsustainable shark and ray fisheries. And well, Chris, welcome to the show. Sorry I took up all your time doing your introduction, but you have a very impressive resume.
Thanks a lot, guys. Thanks for having me. I try to be a jack of all trades, I guess.
Great.
Yeah, definitely.
So to hit you up with our first question here, like, how did you get interested in marine biology and sharks?
Yeah. So I guess I'm probably like a lot of people when I was little, that's what I wanted to do. And I kind of never grew out of it. So my parents say that when I was, I think 3 or 4, I declared that I wanted to work on sharks, and kind of done everything in my career since then to work towards that goal. So I grew up in Hermosa Beach, a little town in Southern California. It's a big surfing town. And we spend our days at the beach, boogie boarding, surfing. I'd see little leopard sharks in the waves, sometimes shovel-nosed guitarfish, bat rays, things like that. And just sort of piqued my interest. And then every once in a while, you'd hear of somebody catching like a young white shark off the Manhattan Beach Pier, and that was kind of a big thing. But yeah, and I just sort of kind of never grew out of it, and I haven't really wanted to do anything else since then.
So we went. So all through like grade school, high school counselors ask you like what you're going to do. You tell them I'm going to go study sharks.
Yeah, I mean, I always knew I wanted to do something in the sciences, but it was always clearly like sharks was the main thing I wanted to do. And so, yeah, every decision I made was kind of towards that goal. And funnily enough, even going to UC Davis, which is pretty far inland, was part of that.
Mm-hmm.
And the reason I went specifically was because they had, you could do a quarter at the Bodega Marine Lab. And so
Cool.
I got my first taste of doing like hands-on independent research when I was there.
Mm-hmm.
So probably one of the most fortuitous things is when I was at UC Davis as an undergrad, I got to work with Joe Check. So he is kind of one of the sort of godfathers of fish physiology. And that's really where I started to get interested in fish physiology. So he let me work in his lab volunteering. So, you start off with kind of menial tasks, washing glassware, data entry, that sort of thing. But he had some really cool projects looking at sturgeon and salmon and how they kind of interacted with a lot of these sort of fish exclusion devices. So anytime there was agricultural setups that were taking water from the delta, they might impinge some of these endangered fish. And so we were studying that. And then working with him, that's when I kind of formulated sort of my first independent research project. So when I went to Bodega Bay, I got to work on leopard sharks, looking at how differences in salinity affected metabolic rate. So I got to build my own flow through respirometer. And I had these really old devices, like old hospital blood gas analyzers that I have to calibrate and then use. And then from there, some friends and I just really loved it. And so we got a little grant through, I think it was the Howard Hughes Medical Institute, to stick around for the summer. And we worked in the eelgrass beds looking at rockfish, surf perches, those kinds of things. And then it just kind of blossomed from there. Once I got a taste of research, that's kind of how it all started, and I didn't want to do anything else. And then when I was looking for grad schools, there wasn't really anyone who was kind of doing ecophysiology of sharks at the time. And I was talking to Joe, and I said, where can I go? And he said, well, there's this guy at Cal State Long Beach named Chris Lowe, and he'd be a good person to talk to. So then I applied to Chris's lab, and then kind of got going from there. So. Yeah, and I've kind of just tried to learn sort of different skills along the way. So, working with Chris, learn everything from like physiology. And then he had a really big, vibrant lab at the time. But it's funny, like there wasn't a lot of shark research going on. There was, so some of the people you might have talked to before, Yanni, I know he was on an episode, Jeremy Baudo, some of those folks were just kind of finishing up. And so when I started, I was working on stingrays, but a lot of people were working on rockfish. And then at the time, there was a lot of work on the offshore oil platforms, which were kind of de facto marine reserves. People weren't allowed to fish on them. And there was this whole rigs to reefs program. So we got to do tons of field work, lots of diving on the rigs, which was really cool. Got to see, obviously, tons of rockfish, pilot whales. One time was one of a gold mine. I did get a mola mola in my transect on the platform. As I was going down, I just sort of stayed still until it got within my window. So the great thing is like we all worked on each other's projects and got to do tons of stuff. And so there was a really like collegial atmosphere. And then from there, yeah, I kind of just wanted to learn more skills, and this job in this chemistry lab opened up. So the lab is IRMES, it's the Institute for Integrated Research in Materials, Environment, and Society, which is why they call it IRMES. It's sort of a mouthful, but it was a core facility for the Cal States. Everything from mass spectrometry, including ICP-MS, GCMS. We had X-ray fluorescence, so electron microscopy. We had stable isotope mass specs. We had laser ablation suite. So we had all these really cool tools. And so my job was to sort of work on any sort of contracts that came through. It was mostly different Cal State researchers. And then maintaining the equipment, obviously, was a big part of it, which is really fun because these are really, really fancy, really expensive instruments, and you kind of have to get over your fear of fiddling with them. And then we sort of, if there was downtime, we could run things that we wanted to, samples that we wanted to. And at the time, Chris's lab was working with the Monterey Bay Aquarium, and we were part of the rapid response team for the juvenile white sharks. This is when that program was kind of first getting started. So we would go out and, we'd tag sharks that fishermen had captured as bycatch. Some of them went to the aquarium at the time, but a lot of them were released. But if there were incidental mortalities, we would take those animals and we'd perform necropsies. And so we had this kind of tissue bank. And I thought, it'd be really interesting to see what the contaminants are. So a friend of mine, Mary Blasius, and we took the samples, and we were running heavy metals and POPs, persistent organic pollutants. And then we found these really, really amazingly high levels of things like mercury, DDT, PCBs. And so that kind of kicked off sort of this interest in toxicology. And then from some of that work, we discovered that basically these sharks could not have acquired those levels of those contaminants by feeding alone. So it had to be coming from something else. And that other source was the mother. So the contaminants are offloaded during gestation. So that was kind of the first evidence of maternal offloading of contaminants in white sharks, which was really cool. And that's become like a burgeoning field. And Katie Lyons, she's sort of taken the torch and run with that. And she's done some amazing work looking at maternal offloading of contaminants. And then again, I wanted to learn more skills. And so while I was there, I worked with a lot of people. I always loved collecting samples of people. And this postdoc from UC San Diego was looking at brains of different sharks and rays. Cara came up. And so I helped her collect some brains from some of the round stingrays I was working on. And then when the opportunity came up to do a PhD with Nick and Cara, I sort of jumped at the opportunity to learn some skills and moved on from there.
So that's kind of an interesting combination. Obviously, we know both Nick and Cara real well, but how did you? It kind of like what those two do. How did this whole thing come about that you're doing brain work with? It just seemed like knowing what Nick does and what Cara does is like, how did that come about?
Yeah, so I think in a former life, or when Nick was doing his PhD, he was really interested in life history and reproductive mode evolution, these types of things. Right. And then I think sort of the idea was kind of spawned by a conversation that he had with another faculty member at SFU, Bernie Crespi, who ended up being one of my committee members. But they knew that maternal investment was really important for relative brain size in mammals. So the idea is that neurogenesis is an incredibly expensive process, and most neurogenesis occurs during early development, especially in mammals. So the more energy that a mother can invest in a single offspring, the vast majority of that energy, especially in early life, is going towards neural development or neurogenesis. So in humans, I think in the really early stages, about 85% of the energy that goes to a fetus is going towards development of the brain and the central nervous system. And it's really cool, in mammals, there are different degrees of maternal investment, but you have they're all essentially within a few reproductive modes. So most of the work was done in eutherian mammals. So it's like us, they have a placenta, but there's similar patterns seen in marsupials. So again, it's just kind of a different combinations or durations of gestation and lactation. But the really cool thing about sharks is that they have every form of reproduction that's been described for vertebrates. So everything from egg laying to placental live bearing. and then this whole range of modes in between. So you have this kind of arena for really different levels of maternal investment, depending on the life history of the animals, their environment, selective pressures, and then also offspring size and number. And Cara had a couple of really amazing papers coming out just looking at the neurobiological diversity of sharks and rays. So she was building on some earlier work from Glenn Northcutt. And what she realized when she expanded the data set is that, we think of mammals having really large brains, and then other vertebrates like reptiles and teleosts having smaller brain sizes. And when I say brain size, I'm referring to relative brain size. That's the size of your brain relative to your body size. Mm-hmm. But sharks have this like range of relative brain sizes that spans all of this. So you have some species which have huge relative brain sizes, things like hammerheads, mobulid rays, and then you have things that have really small relative brain sizes. Typically, there's going to be like squaliforms, skates, things like that. And so again, what's kind of the difference between these groups? Oftentimes it comes down to life history. And a big part of that life history is reproduction and then maternal investment. So they kind of had this idea, and then they were looking for a PhD student. And so I came in and kind of took that initial idea, built on it, looking at the sort of sequence and tempo of evolution, brain, not just brain size, but brain organization, and then spinning that out into a number of different things. So it's really cool because I was really focused on ecophysiology and those types of things first, but then it really made me start to think about the full diversity of sharks and rays. As you both know, there's more than just, everybody probably thinks of like 5 species of sharks when they think of sharks, but there's over 500 species of sharks, 600 species of batoids. There's this huge amount of biodiversity that is oftentimes underappreciated. So I do remember when I was interviewing with Nick and Cara, one of the questions they asked me was, well, what if we decided we didn't want to work on sharks? We actually wanted to work on something else, like a different taxonomic group. So I said, that would be kind of interesting. And there's always a possibility, but I think sharks and rays are actually a really ideal group for these reasons. And I think that in their mind sort of crystallized that, okay, this is the person who can take this idea and run with it. So it's not to say there's anything bad with loving sharks. We all do. But the thing that I really try to champion is that like they're actually a really good group for asking a lot of these really broad ecological and evolutionary questions. And it brings me back to Dave. You'll know this. So I read this paper because you recommended it. It was one of Leonard Campagno's sort of review papers, like alternative life histories in time and space.
Yep.
It's kind of a classic paper that every student of shark and ray biology should read. So he does everything from define ecomorphotypes to the evolution of these things, reproductive modes. But it has, I think, one of the best concluding paragraphs of all time in any paper. And I've sort of shared it like in talks and just like science communication. But the idea is that, people always think, when they think of like groups that are worthy of studying, it's the birds, it's the mammals, it's the reptiles, it's the teleosts. But there's a big number of reasons why sharks and rays should not be overlooked. And they're actually a really ideal group for doing a lot of these things. And so that's kind of what I'm trying to like gear my career towards, is focusing on that biodiversity and why it's important and why we should appreciate it.
Yeah. Just to reiterate to everybody out there, that Leonard Campagno's 1990 paper, Environmental Biology of Fishes, Alternative Life History. It's absolute classic, and any serious student who wants to get into this, that's the paper. It's a must-read paper for you. It's so revolutionary, and just think. You talk about a guy, I've always talked, Leonard thinks outside the box. It was such an outside the box type of paper. It was so revolutionary at the time, and a lot of people don't even know about it these days.
Yeah, it was kind of overlooked. Yeah.
And I've told the story, but I was a grad student when he got onto that whole thing, and there's a book that really kind of helped drive the whole thing too. Is he refers to it in the paper. It was by Robert Backer, who's, if you're a Jurassic Park fan, he's kind of like the actor in the, I can't remember the actor's name in the movie, the original one, but that was kind of modeled after him. and he wrote a book called The Dinosaur Heresies. And
Mm.
that's a phenomenal. I've read that book too. I couldn't put that book down. And that gave Leonard a lot. Leonard was already kind of working on it. And when he read that book,
Yeah.
he just it just inspired him. And I actually got the book too and read it. It was just, those are two kind of complementary things to read. The Dinosaur Heresies by Robert Backer. I think it came out around 86 or so. And then Leonard's paper, if you want to just get inspired to think differently in terms of your research, you're doing shark research. It's just incredible paper. But
yeah, and
I'm glad to hear you read it and you picked it up.
Yeah, I sort of reread it occasionally. I think, like I said, like one of my favorite paragraphs in any scientific paper, it clearly was inspired by that, because I think it's time to nail the list of Chondrichthian heresies to the cathedral door of marine biology.
Yep.
And then it just goes on a short rant, which is really great.
Right. And he talks about he refers to the rise of the fabulous fur balls, which were the mammals that took over from the dinosaurs. He comments on that too, and he acknowledges Backer in the acknowledgments too. But again, anyone listen to this paper, this thing, get this paper. If you can't find it, hit me or Chris up. We'll just direct DM us, and we'll give you the reference. But that's an absolute paper you need to read on that thing. It's just such a good paper to read. Well, anyway, I was glad to hear that kind of inspired you to, as far as your the work you did. I thought the whole stuff you're doing with the brains and all that stuff was pretty cool. I just was kind of curious. I never really asked you how that whole came about, with that connection between Kara and Nick.
Yeah,
and everything.
and I think there's kind of this, I would say, like a kind of like overarching idea, and like, Nick is obviously a lot of my scientific endeavors and ideas are obviously shaped by my advisors and things, but he had this idea, and I think it sort of guides his research now. It's like, let's say you're on your deserted island and you find like this new species of shark. What can you tell about its behavior, life history, ecology, anything, just by what you have in your hands? You can't track anything like that, but you can look at things like the brain, which we can infer a lot about behavior, sensory specializations, energetic requirements. And from that, you can infer things maybe about the habitat, their feeding modes, their behaviors. If you look at things like the gills, can you understand their metabolic requirements? So the idea that your gill surface area is a limiting factor in how much oxygen you can uptake, and also how much waste gases you can release. So the idea is you can really learn a lot about an animal by looking at its morphology. And so I think that's been kind of an overarching thing for a lot of the stuff that he's done. And then obviously, you're always guided by your scientific mentors. Nick and Cara obviously being huge ones for me, but even Chris, when I was doing my master's, his big thing was like, think about everything that you know as like a tool in your toolbox. So that's why I've always tried to learn new skills. And it's, it helps you be a more well-rounded scientist, helps you have conversations with different people, but also I think helps you ask better questions too, ultimately. And then even, what I've been doing more recently. So in my last postdoc working with Aaron McNeil on the Shark Meat project, I, took some of those skills, the taxonomy that I've been working on, the phylogenetic stuff, and put that towards more applied stuff, and then building on like quantitative skills and modeling. So, and then it's really cool to have, research that's much more applied as well, especially considering the current sort of conservation crises facing sharks and rays. So,
Well, I think you've hit on I think a good point that young people listening to is you learn throughout your career so far. You've always made an effort to learn new skills, and I think, and I know you're out there looking for a position now, but those skills are what's going to help you get a position somewhere, more than how much you know about the brain construction of brown stingray or something. And I always tell students, like, like myself or Nick or Kara, we'll teach you about all you want to know about rays and sharks, but the skills are what's going to help you get a position somewhere. So always just learn as many skills like you did. You're one of the best examples I could think of recently we've had on the show, a young person who just like really got a wide range of skills as you've gone along so far early in your
Yeah.
career, and definitely a lot of diversity in that.
Hmm.
So you got through your, how'd you happen, just out of, again, how'd you happen to pick Nick at Simon Fraser? How'd that come about?
So he had just started, or he'd been there for about a year, and so he had must have had like some startup funds, but he was looking for PhD students, and so myself and another guy, Rowan Trebicle, ended up being his first PhD students, and we worked
Mm-hmm.
on really different things. Nick is also a really broad scientist, works on
Right.
all different things.
Yeah, he
He
is.
Yeah,
worked
he
on
is. He is.
coral reef fish before,
Coral.
so
Yep.
that's kind of more what my buddy Rowan was doing, and I was kind of really into the shark and ray stuff.
Mm-hmm.
And it was cool because everyone in Nick's lab worked on something different. So there were people doing life history, people looking at distributions and conservation, and then obviously at the time Nick was I think he was just starting out as the IUCN co-chair. So obviously a lot of students that
Yeah.
came in subsequently worked on that sort of stuff. But it was just a really cool lab where we all learned from what each other was working on. So whether it was
Mm-hmm.
uh, trophic dynamics and trophic pyramids, or age and growth, or neurobiology, or conservation, like everyone's always working on something different. And it was kind of the same in Chris's lab when I was doing masters. It was a big lab. We all helped each other out and worked on different people's projects. I think that was kind of a key to sort of the success in both of those places, was just having people working on different things. So you can always jump into something new, whether it's going out in the field with somebody in Long Beach, diving for rockfish, or diving on the platforms, or whether it was just thinking about people's figures or analyses at SFU in Nick's lab. So there's always something new to do. It just keeps you interested and keeps you motivated, and also I don't know, just keeps everything fresh. So you don't get kind of bogged down in like the one thing that you're working on.
So you've had, go ahead.
No, go ahead.
I say, so you've had quite a array of postdoctoral positions in Canada, the U.S., and Australia. I'm very curious, for people interested, how'd you wind up with such a diverse? I mean, it's in so many different locations.
Yeah, well, I mean, part of it is kind of like, taking the skills that I have and applying them towards different systems. So when I finished up my first postdoc was still working with Nick, but it was part of a larger project with Colin Simpfendorfer and Michelle Heipel. It was Shark Ray MPA project. So the idea is, what can we do to help optimize the design of MPAs for sharks and rays? We still have a couple things in the works that are coming out, but so I came aboard to kind of look at using the phylogenetic comparative approach to look at what things sort of shape and drive the movement activity or behavior of different species. So things like dispersal, so how far animals move from point A to point B, and also activity space. So the idea, if you can understand what drives species needs for space, whether it's foraging, movement, those types of things, how can you optimize MPAs in a particular area? So let's say you go to a new place, you want to put an MPA there. How big does it need to be to protect the sharks and rays, or how does it need to be designed? Mm-hmm. So it was really fun because I really started to, even though I had done a master's in Chris's lab, this is the first time I really got to work with telemetry data directly. So there are some really great movement ecologists that we worked with, Vinay Udiware and Ross Dwyer, and we sort of got to use a lot of the, and this is also where I started to jump into big data. We use data from IMOS, OTN, tagged animals, whether it's acoustic tags, conventional tags, or satellite tags, to kind of build mechanistic models to understand these sorts of things. And we're still working on the research outputs from this, but that was kind of the first foray into that. And then my second postdoc was a much shorter one. It was down in Tasmania, working at UTAS, and the idea was to, again, take that phylogenetic comparative approach and look at life history diversity in penguins. So again, it's a much more, I guess we could call it invariant group. So obviously they're all egg layers. They're sort of restricted range contemporarily. But if you think about sort of extinct species of penguins, especially like the giant penguins, things that got up into South America, further up into Africa as well, there's a huge amount of diversity there. So again, it's taking that phylogenetic comparative approach and just applying it to a new problem. And then from there, that's when I came back to Canada. And this is kind of when we first started, like an idea that had been percolating during my PhD kind of came to fruition. So what you mentioned earlier, Dave, Sharkopedia. So this is an open access database that we've created to provide access for reliable, curated life history traits and population abundance trends of all sharks and rays. So it started out as shark traits. So myself and another PhD student, Seb Pardo at the time, came up with the idea because we were both building our own life history databases, and we realized there's a lot of like duplicated effort here. People are doing this all the time, or we're having to everyone's reinventing the wheel. So if someone's doing their own comparative paper, they're building their own database, they might be missing things. So the idea was to just reduce people's time and effort. Also get around paywalls. So people at some universities might not have access to every paper. So if people can't access Leonard Campagno's paper, we have that available. And so that we started that with support from the Shark Conservation Fund. And then we brought that together with the Global Shark Trends Project, which Nick had, which was these abundance time series. We brought those together into a single database, Sharkopedia. And so it's mostly been supported by Shark Conservation Fund and Save Our Seas, and it's something we're still working on, and we're trying to expand it. Our coverage, we're going to be running a workshop at Sharks International next year, focusing on data from around the Indian Ocean and early career researchers, and just getting other people access to it, training people, and bringing in more data that we don't necessarily have access to ourselves, and also. Even that has kind of developed into something that's more conservation oriented. So you're both familiar with CITES, Convention on International Trade in Endangered Species. So for any species that CITES listed under Appendix 2, those species can be traded, but in order to do so, they have to have a non-detriment finding. And so countries basically have to show their work and say that their fishery is not impacting the population. And a part of that framework that they have to do that is sort of intrinsic biological information. And to make that process more streamlined, some folks at Blue Resources, Daniel Fernando at Akshay Tana, developed the ENDF. So it's an electronic form of this. It makes it more reproducible, rapid, and that sort of thing. And that actually uses Sharkopedia as the backend to build out the intrinsic biological information. So anything that goes into Sharkopedia is now going directly into the NDF process to help the implementation and enforcement of CITES. So we're trying to, just make these things more widely usable and applicable, and increase uptake. But at the same time, just putting the information in the hands of anyone who needs it, whether it's researchers, conservationists, managers, those types of things.
So things like the Sharkopedia and stuff, that's accessible on the internet.
Are you having trouble hearing me, Dave?
Yeah, a little bit. Yeah,
you cut out a little bit, but you're okay.
You're okay. You kind of come and go a little bit. There's a couple places I didn't hear you very much, but yeah, just
okay.
So if I say something that you may have answered, I just we'll work it out. We'll edit it and stuff. But so the Sharkopedia thing, is that
okay?
So is it, I didn't hear if is it web based pretty much. The Sharkopedia.
Yeah. So we have a website. So it's sharkopedia.org. So you can go, you can search by species, you can search by trait or trend. So we have information on age and growth, reproduction, demographics, things like relationships. So length, weight, length relationships. So conversion ratios, those types of things. And then all the population trends are available. And one of the things we wanted to do was build on FishBase. So FishBase is a great resource, but everything is kind of boiled down. So let's say you want to go and look up blacktip shark, Carcharodontis lymbatus. What is the age and growth estimate of it? You're going to get a single estimate of age and growth parameters. But when you do that, you lose a lot of resolution, especially for species that are wide-ranging. Populations might differ. So we maintain the resolution of all of the data. So you can search by parameter, even if you want to search by growth model, those types of things. And then ultimately, everything is traceable back to a resource. So people can use this to freely generate their own data set that they want to use for comparative analysis, for again, for doing things like conservation information or intrinsic biological information of exploited species, those types of things. And then, we ultimately want to expand this with some other information. So some of like the threat information that we're working on. So outputs from like the Global Shark Meat Project and these types of things. So estimates of like fisheries landings per species, trade per species, and commodity type. And ultimately, this is always going to be freely available for everyone. That's kind of the first thing it was built on. And then it's also curated information. So anytime data gets submitted, an editor such as myself or others will check the data before it actually gets imported and becomes available. And so it'll always be up there. People can use it. Sharkopedia.org. If you ever have if people want to submit data, they totally can. Submit their own data if they find if a paper of theirs isn't included. We can provide training. We can provide the stuff to do it. The more data, the better. It'll only improve information that people have to make decisions.
Yeah. So you're going to be doing some of this stuff next year at the Sharks International in Sri Lanka?
Yeah. So we're running a workshop right before the meeting. And, we'll be trying to get as many early career people from around the Indian Ocean as possible because it's a really critically important area for global shark and ray conservation. So, a lot of places have really good fisheries management. The U.S.,
so the goal for
Australia, Canada, for example, good fisheries management, but there's a lot of places that have really high shark and ray biodiversity and don't. And so what they're missing is capacity and information to help them develop fisheries management strategies, conservation strategies. And so we want to provide that for them. And then also build the capacity in those places so they can continue on. So it's not that we don't want to be the only ones doing it. The more people that are part of the community.
Yeah, it's definitely an area of high biodiversity. That's for sure.
Yeah.
You're definitely in a good. So you're definitely in a good area there in terms of high biodiversity and stuff. So that should be pretty good. Well, I'm hoping to be there, so hopefully I'll see. Check it out.
Hopefully,
I'm sure I'll hear from
the Sharks International meetings are always really fun.
In Sri Lanka. Oh yeah, they're like the go-to meeting for me these days. It's like it's only once every 4 years, so it's kind of a good gathering point. And I see a lot of people at those meetings that we don't normally see, like in an AAS meeting or even OCS meeting. You just get a lot of people that are able to go to those meetings. So I think it'll be pretty well. At least the international people who do international work will be there. I think pretty well show up pretty well for that. So
and they're always in really nice locations too. So
yeah, it is.
Sri Lanka next year is going to be a lot of fun. I think it's
exactly
definitely.
It doesn't hurt when it's somewhere nice to go.
I'm sure I'll have Daniel Fernando has been on the show here before. We'll probably have him and some people back next year to talk about probably the Sharks International both before and afterwards. So we'll get some more. In fact, we had Paul Cox and Allie Hood on when they had the last one in 2022. So we'll sure be talking about that. Maybe we'll have one of you or Nick or somebody on to come talk a little more about your workshop stuff as well before or after. That'd be really good.
That'd be really cool.
Yeah, if you're a young person out there, that's definitely a good. If you can afford to get to Sri Lanka, it's definitely the conference to go to.
Yeah, start applying for funding now. I would say for the because I think it's May next year, right? May 2026.
Or work that extra job if you need to.
Sometimes you have to.
Definitely look into that. Check it. So you got the with the Sharkopedia and stuff going on. Obviously, you're pretty busy and stuff now. You're actually on your way back to Halifax now.
Yeah. So driving across Canada. This is gonna be the third time that I've done it. We're going a little bit slower this time, which is taking our time. I teach a class at Dalhousie every summer. It's the biology and conservation of sharks and rays, and it's a really fun class to teach. For the students at Dalhousie, I think for a lot of them it's kind of a highlight. So Halifax is cold a lot of the time. There's not a lot of opportunities to get out in the field, but summers are really nice. And there's a really high biodiversity of things just off the coast of Halifax. So, one of the big components is we do some field work. So we go out and we tag blue sharks just offshore. So we go about 10 miles offshore, And we've seen tons of things out there. So tons of whales, bluefin tuna. That's the first time I saw like a pod of Atlantic bluefin tuna just charging. I thought they looked massive. They were probably like 500 or 600 pounds. And the boat captain was like, yeah,
Wow.
those are pretty big. They get a lot bigger than
They
that.
get bigger.
Yeah, exactly. Last year we saw leatherback turtles, which is the first time I had seen them.
That's
It's
cool.
pretty cool.
Wow.
You know, being in Canada, seeing sea turtles, you're like, what the heck is going on? But
I
you
know,
with
they
the
don't
Gulf,
happen
with the Gulf
often.
Stream, I know.
Yeah.
With the Gulf Stream coming up, it's surprisingly warm. So
Yeah.
you know the water temperature varies a huge amount from season to season.
Yeah.
So in the wintertime, you know the water can get down to about -1. Doesn't freeze
hmm
most of the time
Right.
because it's salty. So yeah, I think I like to surf. Like the coldest day that I surfed out there was -27. And the
Oh,
water was about -1. But then,
Oh.
there's usually once a week every year when you can surf in your trunks.
yeah.
The water will be like up in the mid 70s. It doesn't
Yep.
last long, but
Nope.
it's
Nope.
pretty
It's one
glorious
day.
when it happens. Yeah, exactly.
You gotta take advantage when you can.
Exactly.
Yeah.
Yeah, it's
Have
cool.
you
Well,
encountered
it's
have you encountered any white sharks up there yourself?
I haven't seen them. But I've been surfing a couple times and they've been around. So one time a buddy and I were surfing and it was really foggy and we could hear all the splashing in the water and I made some joke about sharks. And then as soon as we were walking up the beach, this lifeguard came running down and said, "Oh, we just saw a huge fin and we're closing the beach." I thought, "Oh, okay.
oh
Well, so maybe I wasn't that far off." And then just where we were living, so my wife and I were about an hour outside a little spot called Westador.
hmm
And we lived out there because it's like a nice rural area. We had a house right on the water. One of the great things, I had like my own sort of private surf break out front, a little point break.
very
And then
nice.
one day, one of the search tag sharks swam right through there. The next day, I saw half of a seal floating or just washed up on the sand spit right there.
Oh,
So they're definitely
interesting.
around.
Mhm.
We see
Yeah.
lots of signs of them, especially getting into the late summer, fall months. They're around.
Yeah.
I haven't seen. I mean, I've seen a lot more in California and on the West Coast than I have in
Yeah.
Nova
Of
Scotia,
course.
but there's probably
Yeah.
more around there. But one thing
Yeah.
I did see last year, which has been on my bucket list for a long time, was a basking shark.
Hmm.
That'd be
So
cool.
oftentimes, people mistake those for white sharks because everyone has white sharks on the mind now. But actually, I was teaching my class, and one of the students, she worked for the Marine Animal Response Society. So they go whenever there's like a marine mammal stranding,
hmm
or oftentimes if something washes up, they collect it for DFO to do necropsies. Uh, and when I was first starting my class, there was this Instagram video going around of this massive basking shark swimming in a bay on the eastern shore of Nova Scotia, and it was in about 6 feet of water. And as I was watching the video, and I thought, I'm pretty sure I know exactly where that is. It's a spot called Little Harbor. And then the last day of my class, the student came up to me and said, "Oh, there's been a basking shark stranding." She said,
Stranding.
yeah. So she's like, nobody can go and get it. So it had apparently washed up on a little island. So I said, "Okay, well, can you send me the coordinates?" And she sent it to me. I said, "It's exactly what I thought it was. It's just this little tiny pile of rocks, basically in the middle of this bay." So DFO wasn't going to get it because they didn't have the time or a boat. Mars couldn't go and get it. So I called DFO and said, "Hey, is it cool if I go?" They said, "Sure." So I didn't have my own boat, but I took my surfboard, loaded up with my dissecting gear. The biggest knife I could find at the time, because I was kind of packing up and moving, was a fillet knife that had, I think, maybe a 5-inch blade on it. So I paddled out there. And so I'm all alone on this island. It's a beautiful sunny day. I just paddle out there on my surfboard, and there's an 8-meter basking shark just washed up. And obviously, unfortunately, the animal had passed.
hmm
Um, so I inspected it. There were no signs of boat strike or boat injury. So what I wanted to do was collect some samples. So I texted everyone I knew that might want something before I went out there. This is kind of a rare opportunity. So if I can get
Right.
samples for people. So obviously I had requests for vertebrae, muscle, liver, all these sorts of things. And then I knew Carl was going to want the brain. So, all right,
Of
that's what I
course.
gotta do. So,
this is a big surfboard to bring all that stuff back.
Yeah. So the shark was mostly out of the water. It was the caudal fin was still in the water. And of course, Nick wants the caudal fin aspect ratio. So
Of course he does.
I took this little 5-inch knife. I have about 3 hours until the tide fills back in. So I set up my phone so it can videotape me and I can yell out the measurements. I take all the morphometrics
nice.
and I start collecting samples. And so I had to get the caudal fin off to measure it. So it was underwater. So I basically had to take this knife, swim underwater, remove the whole caudal fin, drag it up onto the beach. That thing probably weighed 150, 200 pounds. Get it up there, get all the measurements. Then I needed to get vertebrae. So I'm going through the top, cutting through, finally got through, and it took 6 vertebrae in total, but I could only carry 3 at a time. So 3 vertebrae alone was probably about 80 pounds. So I'd get a section out, paddle it back to shore, go back out, get the other ones. And mind you, these vertebrae, the radius was probably about 7 inches. So that just a single vertebrae is bigger than the knife that I have.
Yeah.
So just hacking away. But the best part was the tide was getting close to filling back in, and I had to get the brain. So I quickly got through the chondrocranium, exposed it, and then there was a lot of cerebral fluid in there, so I had to cut a little trough in the cartilage to let the fluid drain out. As I did that, the tide started to fill in. So I had to quickly take my knife and dissect out the brain, and I got the whole brain out in probably about 10 minutes.
Wow.
And then brought it back to shore. And then because it was the 4th of July, I packed the brain in some formalin and took it to the pub to show off to everybody. So
I'm sure that was a treat. People must have loved that.
And I just so people don't know, if you haven't worked at a basket, their skin is really tough.
Yes.
There's a lot of denticles. You must have gotten, well, you were probably wearing a wetsuit, so you probably didn't get torn up too much.
It was warm. So I just kind of just wearing trunks. And then, I was wearing nitrile gloves at first, but it would shred right through those. So then I had to get a pair of mechanics gloves to wear those. But I was my arms were completely shredded. And a lot of sharks, if you go one way, they're rough. If you go the other way, they're smooth. Basking sharks, it doesn't matter. They're just rough every way you go. But it was a really spectacular animal to see. So
what a story.
That's a great story to share with your students or something someday. Just all the going out there by yourself and taking off. And it was the good thing about technology, you can set up your camera just to record everything.
Exactly.
So take the stuff off. And,
I was definitely in my happy
place. Do you still have the video?
So.
Do you still have the video?
I do. Yeah. So, and we're still trying to figure out what happened to the animal, so why it's stranded. So, and one of the reasons I took the brain, not only to obviously study the brain size and organization, was there's been a sort of more recently, especially animals in temperate water, sharks kind of washing up, and there's been evidence of them having a form of meningitis, which could cause some behavioral impairment, and this is why these animals might be dying of stranding. So I'm sending it to a vet to sort of analyze pathologically to see if we can figure out that that's what it was. Because as far as I know, this would be the first case of this meningitis in basking sharks.
Right.
But other laminiform species have had this. So white sharks have stranded with it, and brushers and things like that. So
salmon sharks, we get them here. We get salmon sharks, which you would know, Chris. So we get them here. These little sort of about 3 foot long salmon sharks wash up. Kind of from about July into the fall, and we always get these calls about a little white shark washed up in the beach. People think they're little white sharks. And we got to a point where I think we must have had like 30 or 40 of these things around. So it just got to be like, send us a picture, because you can only you get so many of those small ones. They're just you kind of know what, once you figured out what happened with them with this meningitis thing. You don't need any more. You don't need any more. So it's kind of like, we're done. We got
it. For sure. For sure.
I think that was one of the first calls I went on with the rapid response team at Long Beach. The fisherman was like, I got a small white shark. It's kind of funny looking.
Yeah.
I thought, it's probably going to be a salmon shark. And that's exactly what it was. But, he caught a salmon shark off of Malibu. So.
Yeah. Yeah,
That doesn't happen often, I'm sure.
No, it's
yeah,
pretty rare.
yeah. Down, we get them down to Monterey. They're not uncommon, but south of
Hmm.
us, we get, especially down to Southern California, they're not that common to
Do they just
wash
get
out.
caught in the current, the Alaskan current?
No, well, they come down. The little
Yeah.
ones come down the coast.
Oh, do they?
Yeah. So,
Yeah.
yeah. So there's been some cool work done by Aaron Carlisle at University of
Yeah.
Delaware. So with a lot of these animals, when they're smaller, they don't have quite the capacity for regional endothermy when they're younger,
Okay.
and so they tend to be in these nursery areas which are further south, and then as they get bigger, obviously their diets change and the habitat changes.
Yeah.
But a big part of that is their increasing capacity for regional endothermy, and they can move into colder water.
Oh, cool.
Yeah.
Okay.
Yeah.
Yeah. That's pretty. You guys didn't Chris do a little bit of work on those down when you were down at Chris Lowe's lab? Did he do a little bit of work on those?
On the salmon shark.
Yeah. Did he do a little bit? I can't remember.
Well, the only thing I remember is that one that we caught that one day. He may have. Oh, he had some students that were a little bit after my time that were doing some stuff up in
Okay.
Alaska.
Oh, okay.
And so
Yeah.
that might have
Yeah.
been.
Well,
Post
Aaron Carlisle came out of my lab or
Yeah.
our lab here at Moss Landing. He worked on
That's right.
leopard sharks, and he went to Stanford and did his PhD, and then that's when he really got into the salmon sharks. And so, because Hopkins is close by us, we were we get the call as Aaron was the guy keeping them. We're saving them for Aaron, or he'd go pick them up, and then once he hit about 30 or 40, I was like, okay, I think we're cool here. We got enough of these things. Picked up here. So they look kind of cool when you first see them, but like I said, you kind of get to where you lose the enjoy or the wow factor kind of fades after a while on them and stuff.
Yeah, I guess that's one of the hard things, but you never want to lose like the joy of seeing these things.
Yeah, and you don't want to. The other thing too is 'cause a lot of times they're found by people in the public, and you don't want to disappoint them because you may find something else later on, and you don't want them to think well they weren't interested in that 3-foot salmon shark, but that 8-foot white shark that washed up on the beach, they might be interested in that.
Everyone's always interested in a white shark.
Yeah, exactly. So that's why you want to make sure you keep everybody happy on that stuff. So we'd always oblige them and go get them and stuff when we can, assuming they're within a reasonable area to go pick them up. It's all part of the citizen science. It's all part of citizen science. It really
yeah. And I think it's a big part of like opportunistic sampling, which I think for as a comparative biologist, that's a really important thing. So like when I was doing my PhD, it was really similar to yours, Dave, where you were just traveling around and collecting samples. So, to get brains, I traveled all around Australia, basically working as a decky on all different kinds of fishing boats. And my pay was in samples. So I worked on longliners, purse seiners. I did go out with researchers as well, gill netters, anything. So, oftentimes these things would come up as bycatch. They'd already be dead. But for me, that was what I was there for. So I was able to get a huge number of samples and a ton of species from doing that, and also just see some really cool things and travel to some really cool places. So
yeah. Well, so if you had kind of going back probably going out with some of the commercial fishers, you probably saw a lot of cool stuff because they're out there all the time. Compared to the occasional survey, the surveys are cool, but you don't probably get as much opportunity as you do on the going out with the commercial guys, which are out there all the time.
Yeah, the thing about fishermen is they know where to fish. They know where they are. So you're much more likely to get a lot of stuff that way. So, and I think, it's also like there's a huge amount of knowledge there that oftentimes doesn't get appreciated in academia. And so it's refreshing to learn from those people, and also just, especially younger fishermen, I find, I don't know if you found the same, but they tend to be much more like keen and interested and even conservation oriented. And so it's really kind of refreshing to talk to fishermen like that, because oftentimes overfishing is the biggest thing driving the extinction crisis that sharks and rays. But a lot of fishermen want to know more and do better, and they are really interested in it. So it's really cool to go work with them. And then obviously sometimes you get your your crusty old guys that aren't super happy to see you. So you see both sides.
Yeah, well. Yeah. Oh, definitely. So so you're bouncing all around, Chris. So what's your ultimate goal? Where are you looking to land career wise right now?
Yeah. I mean, kind of. I'd say sort of like two main things. So I know I've always been keeping an eye open for like an academic position. So one thing I really enjoy is teaching and especially mentoring students. So mentoring
Mm,
undergraduates and graduates.
mhm,
And I think, kind of a really ideal place for me would be somewhere like the Cal State system. So,
mm,
they're generally universities that only have like a master's level students, but you can do some really amazing research at that level.
Yeah.
You also get to do more teaching and kind of mentoring. And I'm a California native. That's kind of what I
Hmm.
love. So I'd love to come back to California, but even being on the West Coast is really nice.
mhm,
I was kind of getting really into the idea of working for government. So there's a lot of really cool research you can do at NOAA or
Mm.
DFO in Canada.
mhm.
Um, for obvious reasons, that's less of a possibility these days. So
Maybe not DFO.
Well,
Maybe DFO has a little bit more opportunity.
well,
Yeah.
yeah,
Chris Lowe recently retired. Are you
yeah,
looking
oh
at his
yeah.
position?
I would love to take over that role. I think there's a really kind of important legacy there,
Hmm.
and history with the Shark Lab.
Yep.
So I think that would be for me that would be like a really ideal fit just for the type of research I'd like to do, teaching and mentoring, and also it's kind of my home.
Yeah,
That's where I'm
for
Yeah.
sure.
from, and being somewhere where I could also jump in the water and go snorkeling or surfing every day is definitely a
Yeah,
plus.
not a bad
So
not a bad
Back,
idea.
yeah,
back to your Baywatch roots.
Yeah,
exactly. Go surfing where it's not minus 30 would be nice.
Yeah.
little change, a little different.
Yeah.
Yeah.
Yeah,
Yeah.
so something like that would be really ideal.
Yeah.
But I think you also have to be just really flexible. I think that's part of like learning new skills and thinking about new
Hmm.
things. So one thing that
Yeah.
some friends and I
Yeah.
have done recently, this came out of an AES meeting last year. We formed this group called MISO. So it's the Mid-Career Elasmobranch Scientists Organization. So this was started by sort of a group of mid-career folks. So myself, uh, Kara Yopak, Andy Milsel, Davi Kasif, Kyle Newton,
Hmm.
and Simon Dedman. And the idea was to sort of build a network for mid-career people who are, kind of
Yeah.
in that postdoc, maybe early professor stage, building some community and some skills. And so we're all trying to support each other in finding positions, finding places to do
Mhm.
research, or even like other endeavors that people are interested in. People have different skills from video production, like cinematography, event planning, music, art, those types of things. So kind of like incorporate those things into the shark world. Um, and just kind of, building a community where we can kind of support each other and get people to the next career stage is a big thing. So,
Hmm.
Nice.
Oh, cool. Cool.
yeah.
So where would you now? Even though you're out there still looking for your sort of job, your niche out there, what kind of advice would you have for a younger person, looking to get started in the field?
Yeah. I mean, I guess kind of like one thing that I've done is especially earlier in your career, focus on building skills. So you know, I think oftentimes when we go through academia, people are trained for an academic career. So part of it is thinking about what career you might want. If you want to go into government, it's a different skill set. If you want to go in academia, it's a different skill set. But the more things you learn, the better. And it's ultimately going to make you a better scientist, much more well-rounded. And also, we really kind of focus on our taxonomic group. We all work on sharks and rays because that's what we love. But don't be like closed to other opportunities. So, my second most highly cited paper is on heavy metal in children's tooth enamel that has to do with autism and other spectrum disorders. So, kind of be open to these opportunities. But I got on that paper because of a skill set I was learning with laser ablation and ICP-MS and those types of things. And so I think that's a big one. Also, if you really want to pursue like a scientific career, like just a perseverance is a really important component, and sticking with things. So like one of the things I'm probably most proud of is one of my PhD papers I had published in the American Naturalist. And so this is a journal that I really highly respect. Um, and this was a paper looking at life history, ecology, and brain organization of sharks and rays. And to get this paper published, I think I submitted it 15 times to 5 different journals. Sometimes it went through 3 rounds of review and was rejected.
Wow.
Sometimes it was desk rejected. And it was a combination of things of getting stuck in this kind of like really niche neurodevelopment argument kind of thing. But the paper that was published and the paper that I first wrote and submitted the first time are almost the same paper. Um, it was just kind of, pushing back against reviewers and like not losing hope because it was something I really believed in. And I also, I had the encouragement of Nick and Cara as I was doing this. So like really pushed through. So, don't let little setbacks discourage you. If it was easy, we'd all be shark biologists and we wouldn't be worried about job security. So, if you're really passionate about it, you're going to have to be willing to, put your heart and soul into it sometimes. But then there's lots of perks to the job. So we get to meet great people. We get to travel to cool places. I've gotten to, travel and fish for sharks and then also surf all over the world. So yeah, there's great things that come from it. So.
Yeah,
that's fantastic.
Yeah. That's great.
Mm-hmm.
That's awesome, Chris. Well, when you get a job, we'll have to have you come back and tell us about. We'll have a celebration with some adult beverages on air for you.
Absolutely.
So definitely keep us posted, and thank you so much for coming on and sharing your story. We really really enjoyed
thanks so much for having me, guys. It's really flattering, considering some of the people that you've had on the podcast are really highly respected kind of luminaries in the field. I think it's really important for the younger generation to hear from those people. So to be included amongst that group of people is a really nice feeling.
Yeah.
Thank you very much.
Yeah. Well, it's definitely deserved, and everybody we've have on is like serves a different purpose, and they all like the goal of this podcast is to inform everybody of what's going on in the industry, what's going on in the field, and how people are succeeding, some of the challenges that they're facing, and being a part of it, and just showing that passion, sharing that passion with everybody, and that's the fun part to be able to witness here at each and every episode. So it's been great. To get to know you and your career and all the fun things you've been able to do, including dissecting a basking shark on a surfboard.
That's right. I was in my happy place.
I bet.
On a surfboard covered in shark guts.
Yeah, you can't go wrong, right? You can't go wrong.
Exactly.
Oh, good. Well, if I don't see you before, I'll definitely plan to see you in Sri Lanka next year. So
definitely.
Definitely.
Awesome.
So
awesome.
Cool.
Awesome. Well, thank you very much, Chris. We really do appreciate you coming on the podcast.
Cool.
Thanks so much, guys.
Thank you, Chris, for joining us on today's episode of. Excuse me. Whoa, I swallowed the wrong way. Let me do that again. Thank you, Chris, for joining us on today's episode of the Beyond Jaws podcast. It was great to hear those stories. It was great to hear your path. Dave, what are your thoughts on this incredible human being?
I think, Chris is like the definition of perseverance, and that's in this field. You have to have that if you think you're going to try to go anywhere in this field, just because it's not easy. There's never been, people talk today about how, well, there's not like a lot of jobs around. Well, I can tell you, going back 40 years, there wasn't a lot of jobs around when I started, and the people before me would tell you there wasn't a lot of jobs. So it's never been a field that there was a lot of jobs available in, and most people eventually go in a different direction. But to stay in it, you've got to have perseverance and a lot of drive, and Chris certainly has that, just by evidence of all the different postdoc he's done in different countries, different continents. But that's what it's going to take if he wants to stick with it and hopefully get a job, whether it's in academia or working with a government agency and stuff. It's just that type of stuff. Even if he got into academia, there's no guarantee he's going to really be able to do much in the way of sharks either. So it's really a field of passion. Again, Chris kind of epitomizes that, and his story certainly shares that. He's certainly a well regarded young researcher, up and coming person in the field.
Well, and you mentioned drive. He literally drove across Canada to
Yeah.
do a course at Dalhousie University that he does every summer. He's done for the past few summers. That's a long drive, folks. If you don't know Canada, or you don't know how long it is, going from Victoria, one side of the country, to the next, is not something you do in a day. And
Yeah.
there's a lot of adventures that come across that. He was telling us of some of those adventures. I don't know if he said that on air. I'm trying to remember if he said it on air, but
Yeah.
let's just say they had some car trouble
Yeah.
halfway through when they got actually not really halfway through, just not even a quarter of the way through. Canada's a big country and it takes a while to get over there, and hopefully he makes it and he's on his journey there by the time this episode he's there by the time this episode publishes. But you know, it's something that you go through, and the great thing is you make the most of it in these journeys. You know, driving across Canada, you stop off, you get to see a wonderful country. Obviously I'm biased because I'm from Canada, but regardless of what you do, is you have to enjoy the moments that you're in. You have to be able to live in that moment, take advantage of those moments, and do what you can. One of those stories is the basking shark story. You know, DFO was not able, Department of Fisheries and Oceans in Nova Scotia, in that story, was not able to pick up, you know, the basking shark, which would probably made it a lot easier for him to take samples. So he takes his surfboard, paddles out there with the tide coming in, and he's got 3 hours to do all the work. He said he shredded up his forearms because, you know, a basking shark skin is not the smoothest. No. You know, we're spending a lot of time covered in shark guts. And let's be honest, in the water with a bunch of shark guts there, that could attract a few critters, you know,
more sharks,
put him in some danger. And luckily he was okay, but he did it for the science because he's interested in the science. He's interested in sharing the data. He mentioned Kerry Yopak that wanted the shark brains and other things, you know, and taking the caudal fin for, I think was it Nick Dulvey or somebody. These things are not easy to do, and he did it because he was interested in the science. He wants to see the science persevere, and so he thought of all the people in his network, all the other scientists that he works with, and he's like, oh, Kerry would really love these brains, or somebody would really love the caudal fins, and somebody would really like this, and I would really like this. And they make the most of an animal that, you know, has passed away, and they use it for science. And I think that's really the definition of perseverance when we talk about Chris here.
Yeah. Oh yeah, definitely. He, you know, like I said, it's not easy working on a shark like that, and it's not easy, you know, moving around all the different locations he's had to go to, especially when he has a wife that's got to be very understanding to be able to be able to do that, and put up with that, I guess, be another way to explain it. And but yeah, no, and I've known Chris for quite a while too, because Chris is originally, as we talked about, was from Southern California, and like I always joke with him, as sort of like, you know, Baywatch of the shark world. And he came out of Chris Lowe's lab for his graduate degree. And we've had a number of Chris's students on here as well now as well. So it was very a testament to Chris's program that he has at California State Long Beach, which I guess he's retired now. And Chris is looking, and Chris Mull's looking for a job, which would make a nice fit because Don Nelson, who was the original person to set up the lab, Chris Lowe was his graduate student that went on to do a PhD. And when Don passed away, Chris Lowe took the job, and now with Chris, thankfully Chris is still with us, retiring. Maybe Chris Mull or one of his students like that could get the job to kind of continue the legacy there. That'd be very cool.
Absolutely. And look, we're not trying to influence Chris Lowe in any kind of way. We're just saying that this could be a good thing that might happen to just fall in the lap. And let's just leave it at that, because we don't, you know, we just think it to be a good idea. That's all, Chris. That's all we're saying.
Yeah. I mean, Chris, we won't mention the fact Chris Lowe's been on our program 3 times now on the podcast. Doesn't make any difference.
But, no.
Yeah, it's part of the thing. So yeah.
But no, it's great to have Chris on always. We can't wait to hear more of some of his things that he's been up to over the next year. We'll have him on definitely upcoming, and maybe next year or so, and to hear more about how this course went, and of course what he's been up to. So it's going to be a lot of fun to look forward to that. So we'll put up any type of contact information for him in the show notes. And of course, Dave, if people want to get a hold of you, how would they do so?
You know, through my social medias, Instagram, Lost Shark Guy, YouTube channel. Please go to Lost Shark Guy and check out the series Searching for Lost Sharks, Extinct or Alive, and please subscribe. And you can also hit me up and Andrew through our Beyond Jaws podcast, our YouTube channel, and on Instagram.
Perfect.
So.
Yeah, absolutely. It's going to be a lot of fun. And I want to thank Chris for joining us, of course. And Dave, it's always a pleasure to continue to do these podcasts with you. And to our audience, thank you so much for supporting us each and every episode when you watch. We really appreciate it. We'd always love to hear from you and your feedback. But until next time, this has been another wonderful episode of the Beyond Jaws podcast. Thank you from Dave and I. We really appreciate it. Have a great day. We'll talk to you next time, and happy conservation.